Today, oh, we don't use cursive, Joe. We print. Today, lesson 2.6, dihybrid crosses. And I know I already have that up top, but something about having a typed title for a handwritten note that stresses me out. Can't handle it. All right, what's this phrase mean? A dihybrid cross. Dihybrid cross. Two hybrid cross. All right, good. Di means two. Good. What else can we glean from this?
It's a hybrid.
A hybrid? What's a hybrid?
It's two things that are...
Yeah. You have a joining of two things put together, right? You think of like a hybrid car. It's got an electric drivetrain. It's got an internal combustion drivetrain. Two of them put together. In this case, a dihybrid cross is when we cross and we're observing two specific traits. Now, to be clear, when you cross two organisms, by the way, what's it mean to cross them? Not to be indelicate, but they reproduce, right, sexually. So when we talk about crossing, we're talking about having two organisms reproduce. So in this case, we are going to observe two traits. Now, when two organisms reproduce sexually, they cross all of their traits, right? I mean, that's how reproduction works. You get one copy of every gene from, the female, from the maternal, whether it's a plant or animal or fungus or whatever, and one set of every genes from the paternal side. Right? But, so when we talk about dihybrid cross, we're not talking about the fact that we only combine two traits. We're just talking about what we look at, what we're observing, what we're tracking. Previously, we did monohybrid crosses. What tool did we use to track the traits that get passed on in a monohybrid cross? A Punnett square. So today we're going to look at doing a similar thing, but instead of just looking at one trait, just at two. Basically, and we're going to talk about it. No, it's called a Punnett square. But it's a bigger one. If you want to call it that, I totally support that, and I might even join in. Okay, so we can observe more than one trait. When organisms reproduce sexually. And we can use a Punnett square to make predictions about two traits at once. Oh, I'm having a moment. Can somebody Google Punnett square for me and make sure I've spelled that correctly?
You did it right. Yeah.
Did I? Yeah. Why does it look so wrong to me today? Do you ever do that where you just write something and all of a sudden you look at it and it looks like it must be wrong?
Yeah.
I don't know what it was.
I hate that feeling, and then you second guess yourself and then you change it and then you look silly. Anyway.
And then when you change it, it's wrong.
Yeah, I have done that a lot. Oh, I should have the word make. That's what I should do. I thought the word make. We can use a Punnett square to make predictions about two traits at once. And we call this a dihybrid cross. Who was the first person to really study modern genetics? We call him sort of the father of modern genetics. Russian fella. Do you remember? Gregor Mendel. Good. What did Mendel study? Very good. So that's what we're going to look at here. Let's talk about the pea plant. Oh, I like that better. All right. So if you remember, we looked at two different traits. Actually, we looked at a few different traits. I don't think we talked about the second one of these. Now I can't remember. But we're going to look at two traits right now, a dominant trait and a recessive for each. So these are ones that exhibit dominance. And the first we're going to look at is seed color. And you should remember this. What does the Y stand for? Big Y? Yeah, the dominant trait is yellow seeds. And the recessive trait is green. Very good. And then, this is one I don't think we looked at, but you will sometimes buy green peas and they will be perfectly round. And then sometimes you'll buy seeds and they look wrinkled. You ever seen that? It's not just that they're dried out. In fact, seeds start off dried out, and some are nice and pretty close to perfectly round, and others are kind of wrinkled. And that is controlled by a gene with two alleles. So now, here's the thing. You can actually have a lot of different combinations here, can't you? Because how many copies of each gene are you going to have? 2, one from mom, one from dad. When you create a sex cell, how many of those will you pass along? But of these 2 genes, you're going to have 2 copies of the seed color gene. You're going to have 2 copies of the seed shape gene, but you're not going to pass 2 copies on to your children. You're only going to pass on 1 of each, right? Because that's how creating sex cells works. What's the genetic process that creates sex cells, by the way? Meiosis. Right? So let's look at an example here. We're going to do a dihybrid cross, and we'll choose together, let's say we have a mother plant who is, where do we want to go here? Let's talk seed color first. Do we want mom to have yellow seeds or green seeds? Okay, yellow. What are the 2 ways that mom could have yellow seeds? Good. She could be homozygous dominant or heterozygous. Which one do we want her to be? Okay, mom is heterozygous. What's that look like? 1 dominant. Y. 1 recessive. Little y. I'm going to make that even more obviously little. And then let's talk seeds. Do we want mom to have round seeds or wrinkled seeds? Wrinkled, which means what would the genotype have to be? Okay. And this is how we would write that. So let's break it down. Why are there 4 letters there? Well, because now we're not tracking 1 gene, we're tracking 2. So these are the 2 alleles she has for the seed color gene, and these are the 2 alleles she has for the seed shape gene. We have to track them both. So she is heterozygous for color. And how would we describe her for seed shape? Homozygous recessive. For shape. And just so we have it clearly then, what is the shape again? Wrinkled. And what is the color again? Yellow. Does that make sense? We're okay with that? And I'm going to highlight that in green. Whoa, what happened here? No, thank you. I'm going to highlight that in green so we remember that that's mom. And then we've got a father. And again, this is kind of silly because we're talking plants here, but it is still the case that one contributes pollen, the other contributes a seed, so male and female. And what do we want the father to be, yellow or green? Green? What's the genotype going to be then? Got to be homozygous recessive. How do I write homozygous recessive? 2 small y's. Good. I'll write that. Oh, go away. Thank you. And by being homozygous recessive, what color are we? Green, good. And we want to go round or wrinkled again? Round. And 2 ways to do that. Which do you prefer? Homozygous dominant, which looks like what? Very good. And just remind me what shape that is? Round. All right. That's a lot of writing, I know, but it helps us to use those terms a lot.
Yeah,
I mean, you don't need to write these over and over again, but I think it helps. It helps it stick in our brain because we got words we got to learn, right? So now we've got a mother, we've got a father. So there we are, and we're going to cross these two. But keep in mind now, when we cross these two, we're not just tracking sex cells with 1 gene. We have to track what kind of sex cells can get made with 2 genes. And that's going to complicate things. How big was the Punnett square last time? 2 columns and 2 rows, right? Because you either got the big Y or the little y. The problem is now, I'm either going to get the big Y with that little r, or I'm going to get the big Y with that little r. Or I could get the little y with that little r, or I could get the little y with that little r. See what I mean? How many possible ways can you combine those 4 alleles to get a sex cell that only has 2 alleles, 1 of each? 4. Exactly right. Good. Look at that math. That was great. So we're going to need not a 2x2 grid, but a 4x4 grid. And you're going to see that, if you'll pardon my language, it's a bit of a pain in the ass. It's a bit of work. But we're going to do. I know you were shocked, Fronan, but try to contain yourself. Ronan's never heard speech like this.
I haven't.
He's appalled.
Parents raised me different.
Well, here's the deal. I think when I think of bad language, I think of language meant to hurt people. You know what I mean? Like that. To my definition, that is what bad language is. I don't think there's bad words. I think there's those words that can be weaponized, right? Yeah. I have a feeling that nobody is going to find my saying pain in the ass hurtful. And if I'm wrong about that, please tell me.
Well, it hits kind of close to home when I was little.
Did you hear that a lot? I think this grid's tough. I think it can take it. All right. Yeah, I do have the advantage of having a digital ruler.
I just try and go over the lines.
That's a good trick. And you know what? I've got extra space at the bottom, which is going to bug me, but I'm not going to do the thing I do where I erase the whole thing. Is what I'm saying right now, but I totally want to do the thing where I erase it, so I'll... Oh, I can't do it. I can't do it. I got to erase it. I'm sorry.
I got you. You're good.
I just can't handle it.
What is math do with
I don't know when in my life I became fussy. I wasn't always fussy. And I've become it, and I don't know why.
So how many columns? 4?
Yeah, 4x4. Okay, that's still not perfect, but we're going to pretend it is. Okay. So 4x4. 1, 2, 3, 4. 1, 2, 3, 4. All right, a 4x4 grid. And we are going to dedicate the top of this grid to all of the possible sex cells that the maternal plant could produce, which would be all of the seed cells. And we're going to dedicate this side to all of the pollen cells that the father plant could produce. So now, if we write this down, we're going to do big Y, little r gets crossed with little y, big R, big R. That's what this table is. If we just want to remind ourselves what we're doing, have it over there. So the question is, when we go to make sex cells out of big Y, little y, and listen, you don't have to draw this, but I find it helpful sometimes. I have old man eyes, and I find it helpful sometimes to have it just big. Does that make sense?
Yes.
Because I, you know. So now listen, I can either have Y end up with R, in which case the sex cell I would get would be big Y, little r. Or this could end up with that, in which case I would have what? These two end up together when random assortment happens, and I would have, yeah, big Y, little r. It is really unhelpful to say YR because all of these combinations will be YR. What about this combination? YR.
Little y, little r.
Very good. Little y, little r. Or we could get this combination here, which would be
Little y, little r.
little y, little r. Okay. Does how we got these four possible combinations out of this make sense to you? When random assortment happens during meiosis, in other words, when we have the cell and we've got the chromosomes lined up, you know, the big Y could go this way and we could get it, or the little y could go this way and we'd get it. Does that make sense? And it's a 50-50 chance. But then when that goes, what about the other chromosome? Well, we could either get the maternal version or we could get the paternal version. So it's just random chance. And when you randomly put them together, you'll either get this guy ending up with that guy, or you'll get this guy ending up with that guy, or these two, or those two. And it is random which combination you get. They're all equally likely, is what we're assuming for now. Not always equally likely. All right, what about the paternal? This one should be a little easier for us, yeah? Little y, big R, big R. So what are the possible combinations? Yeah, no matter what you do, you're going to get little y with big R, or you're going to get little y with big R, or you're going to get, okay. So, not the most exciting combination, but does everyone understand why all four of these are going to be the same? That's just what you chose here. You chose a homozygote and a homozygote, so we get little y big R, right? Little y big R, little y big R, and little y big R. All right. And now we can actually do the cross. Again, I just kind of do that as rough work over there. I find having a little bit of rough work helps. So now, if this seed cell happened to get pollinated by that, what would the genotype of the offspring be?
If those two.
Yeah, if these two here join up. What's that combo? Well, big Y and...
Just take the letters and put them in the box.
Yeah, that's right. So we have a big Y and we have a...
And the little y, and then we got the little r and the big R. But
yeah, and by convention...
They're all going to be that. It's just the order that you switch. Really.
They're not all going to be the same. We're going to have a lot of the same. So look, is this whole column going to be the same?
Yeah.
Yeah, so this is a nice easy one. This whole column is the same. And the next column is actually also the same.
I feel like we're not organizing much because of that. I don't know.
Yeah, this particular one, is pretty straightforward, which is nice. It'll be quick to count. Some of them are pure misery to count.
Yeah.
We'll do one of those next, don't worry. This one will be quick. So this whole column is going to be the same, isn't it?
Yes.
Big Y and a little y. Big R and a little r. By the way, by convention, we usually do uppercase then lowercase. Right? We don't try to sort it by mom versus dad. We just, if they're both, we always say uppercase then lowercase. It's not a hard and fast rule. I don't care. If you want to switch it, it's not wrong. But that's just what you'll see done. So these are all the same. All right, that was fairly easy. And what about here? Very good. And will this whole column be the same? And in fact, will this one as well? So we actually only get two possible outcomes from this particular cross. That wasn't so bad, was it?
Now the Y's aren't looking like Y's. Too many of them.
I'm doing my best. Oh, you mean yours?
Mine.
Okay, because mine are looking pretty sketch too. But that's okay. So, now, what are the possible genotypes you could have? I don't mean from the table. There's only two genotypes on the table. But what are all the possible combinations you could have? Can you think of them? What could you be? I mean, you could have big Y, big Y, big R, big R. That's one. How many of those did we get? 0 out of, how big is this grid? 16. Very good. You could be big Y, little y, big R, big R. How many of those did we get? What else could you be? Give me the next one. We could be small y, big R, big R. How many of those did we get? Okay, what's the next one we could be? Small y, big R, small r. How many of those did we get? 8 of 16. What's that as a percentage? Good. What's the next possible combination? Yep. Small y, small r, small r. How many of those do we have? 0 of 16, which is 50%. And finally, what's the last possible combination? Did I say 50? No, I didn't. Sorry, what was that? Looks good to me. What's the next possible combination? Big Y, small y. Yeah, small r. How many of those do we have? None. And what's the next combination? Big Y, big R, small r. None of those, right? Oh, yeah, we did have those, didn't we? And is there any other combination we haven't thought of? 1, 2, 3, 4, 5, 6, 7. I think there's going to be an 8th one. What's the 8th one? Oh, no, is that it? I think that's it. Do I have a duplicate in there?
Yeah, and I didn't want to write it because it seemed like.
Which one did I duplicate?
You've got like 3 or 4 duplicates there. Maybe 5.
Do I? See, this is confusing, right? Yeah, well, listen, you'll succeed. I'm easily discombobulated, and I like to think from time to time I'm combobulated. I have my moments. Yeah, there should be 8. I'm missing one, but I can't spot which one I'm missing. It's easier when you actually have them, right? Because it's easy to see that these ones are all the same, right? Yeah, that's right. That's the one we don't have. We didn't do big Y, small r, which we happen to have none of. There we go. So you can see here we only actually got two outcomes.
Big Y, big R, small r is 0 out of 16 and then turn to 8.
That's an 8, as you can clearly tell. Quiet you. Somebody corrected me, and instead of erasing it, I just said it equals the other. I'm doing my best. It's the best I can do. There we go. I'm really glad I'm recording this. It's going to make me look good. And then the other combination we had.
Didn't we already have that? I'm not even.
I appreciate that. All right, and there we are. So we only had two outcomes that we actually observed, but I think it's useful to talk about the ones we could have seen, right? Does that make sense? So in this particular cross, you only get a couple outcomes, despite the fact that there's lots of outcomes you could have had. Those are the genotypic frequencies. What are the phenotypic frequencies? First of all, what's that mean? We only saw, well, what's this guy look like? This fellow right here, what's he look like? Very good. I'm so glad I highlighted it in yellow, but that's okay. He's green and round. And how many of those did we have? 8 of 16, which is, of course, 50%. It's also equal to 0 out of 16, if you're wondering. And what do these guys look like? What do they look like? Yeah, they're yellow and round. And that's the other 8 of our 16, or the other 50%. All right, now this is only tracking 2 genes, and how much more work are we doing? 4 times as much work, right? Can you imagine tracking the 22,000 genes that a human has, all the possible combinations? Wild, right? I mean, current computers simply can't do that math. That is more computational power required than we currently have available to us, just to talk about one generation of reproduction. Think about it. Think how big that grid would be, right? Be enormous, because it grows by the square of that number, which is, spoiler alert, lots. Actually, sorry, it doesn't grow by the square of that. It grows by 2 to the that exponent, which is real big. Try to put 2 to the 22,000 in your calculator, see what you get. Spoiler alert, your calculator will not give you an answer. It's just too big. Glaring at me, but it's true. Trust me, maybe your calculator. Really? Yeah. You're welcome. Okay, can we try this again? This time you're going to do one. All right, so we just did an example. I'll leave the example up here, but now let's look at example 2. And example 2 is going to be crossbreeding heterozygotes. So let's look at the same 2 traits just to make life easy. And let's cross, I did color then shape as a rule, right? Let's cross a heterozygous mother with a heterozygous father plant. Can you do that? I think you can. I have faith in you. So I was thinking I might leave the example up That way you can kind of follow what we did there. Does everyone have the question written down? Doesn't take too long to write the question down. Oh my. And while you do that, I'm going to see if I can make Excel do it. And do a little experiment here. All right, so how's that looking at that example? Is that helpful? Again, you guys have the question written down? All right. How do I do that again? Data validation. Data validation. List. Source. Hey, get to work. Damn it. There we go. Damn it. Oh, team, I think I did it. Pretty good. All I'm saying.
There should be 9 because there's a 9 below.
You sure about that?
Yep. I just spent like 10 minutes
All
analyzing
like over each other because there was one missing.
It's easier once they actually exist. Anyway. Okay.
Because I was like,
I believe you. I believe you.
I didn't believe Sophie at first until I was ready.
I always believe Sophie.
I didn't.
Sophie told me my name was Mr. Calford. I believe her. Like, all right. Here we are.
Oh my gosh, I see it. Yeah.
Okay.
2 missing. You only have 7 right now.
No, I meant the other numbers. I was writing. No, I added them. I added them in the first number of the 16 to make sure, and I had 14, so I had to recount. And then I got.
Oh, there's so many YYRRs.